human-elephant conflict was a major issue in sub-Saharan Africa, as well as I've learned subsequently, also certainly a major issue throughout Asia as well. Briefly, human-elephant conflicts leads to a lot of problems for both humans and elephants. So with respect to humans, human injury and fatality is certainly a big problem in the context of human-elephant conflict. It's estimated, though the numbers aren't great, the data are not super well available, that approximately 30% of large mammal-caused human injury and fatalities are due to elephants. And elephants are, of course, pretty famous for causing damage to human livelihoods and potentially affecting food security because they tend to like to crop raid. And in the context of raiding individuals' crops, they also do a great deal of damage simply by trampling the crops that don't manage to get eaten. And that's a really big problem, particularly if you are a subsistence agriculturalist, right? There are farms around Kibale National Park in Uganda where we do have a very large elephant population. And it's pretty amazing to see what happens to these fields after an elephant visit, where a family's entire livelihood, their entire food security system for the entire year can be compromised by a brief visit from elephants in a matter of minutes. Humans, as we generally we don't take this lying down, and human retaliation against elephants is a major cause of elephant mortality and elephant injuries. As well, the presence of elephants and the presence of particularly elephants that are doing things like crop raiding or sort of having these negative interactions with humans has been listed by people living in these elephant areas as one of the major reasons that they are resistant to localized conservation schemes. So, for example, in Kibale National Park, we conducted several interviews with folks living around the periphery of the national park and protected area. And what we discovered pretty quickly was that generally folks liked the park, but they did not like the elephants, which caused them to blame the park and generally try to resist local conservation schemes. So human-elephant conflict is a really big problem. And because it's a really big problem, there are, let me just see, sorry, I'm distracted by the chat. There have been several attempts to mitigate human-elephant conflict. Here are the three sort of best known and arguably somewhat effective current strategies. One is scare shooting, basically showing up with a big gun and shooting it in the air to auditorily deter elephants, basically to scare them out of an area. Trenching is something that's been increasingly tried, where literally these very deep trenches that you can see here in this middle photograph are dug sometimes around the entire periphery of national parks and protected areas, with the goal of excluding elephants basically from the non-protected areas by causing them to have to cross these trenches in order to get out. You can see how big of a scale that can get to a little bit in this picture. And bee fencing has also been very, very effective at deterring elephants. What bee fences are, for those of you who aren't familiar, is basically these are these schemes where farmers will set up honeybee hives, basically, and connect the hives with usually some kind of tripwire or something that is basically, if it's moved, it's going to cause the hives to actually move, causing the bees to swarm out. And elephants, amazingly, despite their size and their extremely thick skin, are absolutely terrified of bees. Chances are, because even though a bee sting wouldn't hurt the outside of an elephant if they stung their haunch or their side or something like that, because they do have very thick skin, the inside of elephant noses are very, very delicate, and apparently bees getting into their noses can cause them a great deal of pain. So the way that bee fences work typically is you set this all up, and then the elephant will, in attempting to cross this barrier, will cause the beehives to jiggle, the bees to swarm out, and then are deterred from the area. And these work to a greater or lesser extent, depending on the context and the circumstances. But they're also problematic in various ways. So some of the problems I've noted with the existing successful human-elephant conflict mitigation strategies, in terms of the trenching and the bee fences, they can be quite expensive to set up. So particularly if you're talking about, you know, a situation where you're trying to reduce human-elephant conflict in the context of subsistence agriculturalists who might be very poor and not have access to lots of money, trenching and defencing can be quite expensive, as well as really labor intensive to set up. Maintaining it can also be incredibly intensive. Imagine having to maintain a giant trench around the entire periphery of a large national park, and you can start to imagine the cost and the labor that's required to actually maintain these things. They also tend to require extensive pre-planning, right? So if you are a farmer living in an elephant habitat region, you basically, if you want to use one of these strategies, you really need to be thinking ahead, right? You need to be doing all of the legwork before you actually have any problem in that area with the elephant. And so these things simply don't work in the moment of a conflict, which is, at least for me, when I tend to react to things. With respect to scare shooting, again, that's been shown to be effective in some places. However, the problem there is, of course, that you need a gun, which most people, due to expense, don't have access to, maybe due to local laws, don't have access to. And it's certainly not necessarily something that we want to start doing, is heavily arming folks living in villages around these protected areas, because there can be quite a lot of political instability in these areas as well. And so just handing out large, loud guns maybe isn't the best way to think about it. So as a sensory ecologist, I primarily study basically the senses, so olfaction, vision, and hearing that animals use to interact with their environments. And I typically, as Martin mentioned in my introduction, do this in the context of mutualism. So do this in the context of plant-animal mutualisms. And so when I started to think about this problem after getting to finally get to Uganda in around, I think 2014 was my first trip there, I really started thinking about elephant signals and what type of signals might be effective at deterring elephants, particularly in the moment. So in the actual moment of a human-elephant conflict, in the moment that the elephant is trying to enter the crop field or the village, or in the moment that the elephant is surprising someone. And so as a sensory ecologist, I started to think about which signals might be most salient to an elephant. And because I have eyes, in addition to being a sensory ecologist, I very quickly noted that elephants have these ridiculously large noses, right? So I thought, olfaction. Olfaction might be a way to start thinking about which types of signals might be deterrent in the context of human-elephant conflict. And indeed, elephants are really highly olfactorily oriented animals. So we are still in the world of sensory and chemical ecology, kind of doing a lot of the work to figure out which olfactory receptor genes bind to which different volatile organic compounds. It's very early stages still, and so we're forced to kind of look at this from this very broad strokes comparative perspective. If you line elephants up with the existing mammals and animals generally for which we know how many intact olfactory receptor genes exist, elephants are consistently at the very, very top. So for some comparison here, the animal with the all-time least number of intact olfactory receptors actually currently belongs to the pufferfish. Primates, including ourselves, tend to be around the 300 to 400 intact olfactory receptor gene range. Dogs are about double that with their amazing sense of smell. And then look at elephants over there with 1,948 intact olfactory receptor genes. Again, we don't necessarily know what these olfactory receptor genes are able to bind with in terms of which gene allows a given animal to sense a given odorant. But it's pretty compelling when you look at these numbers, just in terms of being convinced, I think, that elephants are highly olfactorily oriented. And a couple of studies came out in the last year, actually, while we were working on this stuff, that showed even more compellingly that with their noses, elephants can do absolutely amazing things. So Joshua Plotnick and his colleagues in 2019 published a wonderful paper that I would encourage you to all check out. It's in PNAS. And he found that with their noses alone, so using only olfaction, elephants can actually count things. He did this by taking semi-tame elephants and basically offering those elephants a choice between two different containers that had different numbers of sunflower seeds. He altered the numbers of sunflower seeds to offer the elephants 11 different potential ratios of sunflower seeds. And overall, the elephants, without visual stimulus or auditory stimulus or using anything but their noses, elephants were able to identify and consistently chose the buckets that contained the larger quantities of food, indicating that elephants can actually use their sense of smell to count, which is pretty amazing. At the same time, myself and the rest of the research team actually that worked on this project, so Melissa Schmidt, Omer Nevo, and Manfred Ayes, who's also at the University of Ulm, we were using the same elephants that we actually used for this predation study to see whether or not elephants could detect variation in nutritional quality in fruits. And we found that amazingly they could. I'm going to show you a brief video of our experimental setup to give you a sense of what that looked like. Basically, for this study, we used the same semi-tame 5 captive elephants that you'll be meeting in a minute that we used for our predation study. They're at a center called Adventures with Elephants in Limpopo Province in South Africa, and they're trained to respond to different commands. So in this case, what we did was we took one of the elephant's favorite fruits, which is a marula fruit that elephants absolutely love and go crazy for. And we basically put one marula fruit in each of these 2 buckets that you can see on your screen. The elephants were instructed to walk up to these buckets, sniff them, and the marula fruits furthermore were hidden underneath these, I'm not really sure what to call these. They were hidden so there was no ability for the elephant to visually see the marula fruits. They're asked to sniff each one of these buckets, each remember containing single marula fruit, and then they were asked to choose. Upon making their choice, we quickly spirited away the marula fruit that they had chosen, as well as the one they hadn't, in order to assess it chemically, as well as to assess its nutritional content. And I'll just show you what that looks like. So here the elephant is being asked to
Okay, trunk out.
Trunk out, remove the trunk.
And choose.
And now choose which is
Good
preferred.
girl, take it.
Here we whisk them away, and the elephant gets free to continue keeping the elephant interested in further trials. Amazingly, what we found after looking at the nutritional quality of the fruits that elephants were choosing was that elephants consistently were choosing individual fruits as long as those individual fruits had, in terms of sugar alone, were 6% more sugar rich than their alternate choice. So in addition to being able to actually count the amount of food being offered in buckets, as Plotnick and colleagues showed, we showed that same year that they could actually assess even this very slight variation in food nutritional quality. So all of which kind of points to elephants have a very good sense of smell. We weren't the first folks to notice that elephants have giant noses and a good sense of smell. And there have been actually several attempts to assess olfactory signal efficacy when it comes to mitigating HEC. Overwhelmingly, those attempts have been using plant-based odors, particularly chili, with the idea that capsaicin, the thing that gives chili peppers the sense of that we perceive as hotness, basically capsaicin, that compound is supposed to be potentially a deterrent to elephants. Results are not fantastic, I don't think, for the chili pepper studies that I have seen. And as well, the issue, I think, with some of the chili pepper studies might be that elephants are not responding necessarily to the odor. They might, in fact, be responding to the capsaicin, which may have some kind of burning or uncomfortable effect on elephant skin, which is something we'd like to avoid. And someone also tried with bee pheromones quite successfully, actually, to synthesize bee pheromones and use them in a deterrence context with elephants as well. But what we started thinking about was, what about predator odors? Because even though elephants might, for example, be very averse to consuming a chili pepper, elephants also spend pretty much all of their time walking around foraging and smelling plants that are both palatable and unpalatable to them. And so we thought, you know, even if they smell a plant that isn't necessarily a plant they want to eat, is that going to cause them to avoid an entire area? And we thought that it might not. So we started shifting our focus to predator odors specifically, and of course had to first ask this question: are elephants prey to anything? And it turns out they are, despite being the largest terrestrial mammal on Earth. Elephants actually comprise a very large part of lion diets, both in Asia and in Africa, and several studies have shown that. Typically, elephants between the ages of 4 and 15 years old tend to be targeted, but older elephants can also be targeted as well, even though lions have less success in preying on them. And it's known that in some very well-studied areas in Botswana, lions are actually considered to be elephant specialists. So absolutely, despite their ridiculous size, African elephants are prey to Leopanthera, African lions. And so this naturally led us to this question: can we use predator odors to deter elephants, to do something to mitigate human-elephant conflict? And so we started this project about 1.5 years ago, I want to say. So I currently am at the University of Florida. What that means is that I live in Florida. And for any of you who have not yet been to Florida, I'm hoping that you've seen Tiger King, which is a very Florida type of thing, even though it took place in the state next door. Luckily, living in Florida, there is strangely an abundance of places that have large wild felids held captive. There's one about a 20-minute drive from my lab called Carson Springs Wildlife Conservation Foundation. And myself and 2 students asked if we could show up and basically collect some lion feces from within the lion enclosures. So what they did was they had a mated pair of male and female lion who shared a very, very large enclosure. They locked those cats up just off to the side, and me and my students ran around terrified in this lion enclosure, collecting all of the fresh feces that we could find. We ended up basically just pooling the feces that we collected because we obviously couldn't assign it to an individual lion, and we didn't think that intraspecific variation would be significant anyways. So we basically collected these fresh fecal samples from this enclosure, from these 2 lions that were fed a normal diet in the sense that their entire diet was comprised of raw meat. And then we started to analyze the compounds that were found inside the lion fecal samples. So what I did was to take about 0.5 kilogram of feces and place it into inert sampling bags. For those of you who are unfamiliar with the methods of capturing volatile organic compounds in the context of analytical chemistry, it basically looks like this. The sample, in this case the feces, was placed inside an inert sampling bag. We actually use oven bags that you might cook your turkey in over Thanksgiving. Put the fecal samples, in this case, into this inert sampling bag, incubated it, so basically let it sit there for about 20 minutes, and then started the pump. And what the pump does is it pulls the air through an activated charcoal container that traps any external to the sampling enclosure volatiles from being captured. It goes over the headspace of the fecal sample, and then it's pulled into tie-on tubing and basically pulled into XAD cartridges, which are cartridges that contain little beads that absorb those volatile compounds. I then sent those samples to Germany, where they were analyzed by Omer Nevo on his gas chromatograph using thermal desorption and a cold injection method. And then we had a look at what we found. So again, for those of you unfamiliar maybe with gas chromatography mass spectrometry, what you get after you run a GC mass spec is an output that is a chromatogram that looks like this image that you see here. And what we have on the y-axis is actually quantity. And what we have on the x-axis is the time that it takes a compound to basically come off of the coil. This is determined, of course, by the compound's molecular weight, which in turn gives us a very solid clue as to, depending on the time and the intensity at which a compound comes off. The time at which a compound comes off alone actually allows us to identify which compound it is. And here's just one of the resulting chromatographs from the sampling of the lion fecal volatiles. there were 3 compounds of note that we found in these lion fecal samples that were basically not present in controls, and that also haven't been found in great quantity in non-carnivore species. And those 3 compounds are here. It's labeled number 2, dimethyl disulfide. This is a kind of gross, very strong garlic, kind of rotting corpse smell. Different people describe it differently. 13, we have a compound called phenol, and 15 is the compound indole. And all of these were present in the lion fecal volatile samples and not in the controls or in non-lion fecal samples. And that's actually not new either. So Mansourian and colleagues in 2016 actually studying the oviposition preferences of Drosophila on feces, also did some of this GCMS work and did it on lions specifically, as well as all these other different animals that you can see here. And these 2 really big clean peaks that you see in this lion graphic here that are from this current bio paper, which I would encourage you to check out. It's really interesting, are again phenol and indole. So we were pretty sure that we had something when it came to phenol and indole being important components of obligate carnivore species. So then we had to figure out, all right, well, do these compounds, or does lion feces itself, is it possible to use these smells to deter elephants from an area? And so what we did is we went back to Limpopo, South Africa, to the Adventures with Elephants facility there, where again, we had the 5 semi-tame trained elephants. And what we did was we basically set up a system where you can see here, there's a handler giving commands for the elephant to walk down this path. There's also a handler on the other side encouraging the elephant to cross over this. The path itself is blocked on either side by really heavy vegetation to deter the elephants from trying to just turn around and walk off. And then basically what we would do is we would take a string. In some trials, it was a string alone. In other trials, because we actually did this over 2 seasons, in other trials, the string would actually be placed into a PVC tube that had holes drilled into it to allow the odorants to escape. And then what we basically did was these good folks, the elephant trainers, would tell the elephant to start walking this way, and we would measure the time that it took the elephant to cross from 5 meters away from the string or the PVC pipe containing the odor stimulant to get both of the elephant's front feet over, and all trials took 5 minutes. In a few cases, elephants refused to cross entirely, and we simply coded that as the full 5 minutes. Additionally, if elephants hesitated, they were given further commands by their trainers, and they're also offered oranges, which it turns out they absolutely love. So basically, the handler on this side would try to encourage the elephants over this odor barrier that we basically set up by offering oranges, 1 each 10 seconds, and kind of trying to roll them so that the elephant would not be able to just reach over with their trunk and grab them. So I spent about an hour this morning making this clip art, and then it turns out that I actually have a photograph of the experimental setup. This is basically what it looks like. So here you can see in this case, we've got the PVC pipe with the holes drilled into it. I believe this is the 5 meter marker. And then the idea was encourage the elephant across until both front feet are over the odor barrier. So what odors did we include in the bioassays? We didn't want to try just lion feces or synthesized lion feces. We wanted to try as much as we could and all the things we could think of that might be relevant. So we tried with some indirect predator cues. In this case, we used rotting impala meat that was basically scavenged from the center. We also wanted to know if maybe elephants were averse to feces generally, as opposed to predator-specific feces. So we tried, in addition to the lion feces, we tried cheetah feces, domestic dog feces, and then some herbivore feces represented by both giraffe and wildebeest fecal samples. We tried the lion fecal sample directly, so actual feces that we freshly collected, again, from that area, and put it straight onto the string. And then we tried it with our synthesized predator feces. So for that, what we did was we took indole and phenol, which you are able to just purchase from Sigma Aldrich. And we basically combined indole and phenol at extremely low concentrations. So the concentrations we used were 1 part per million. And then we compared everything to odorant controls, which in this case were just water on the string, either alone or in the pipe. Then what we did was we used GEEs to basically assess differences in the time it took to walk the 5 meters and step over odor barriers with both front feet. We also did a little analysis to look at the number of commands that were required from trainers before elephants would cross. And what the GEE does is allow you to compare all of them together. So before I show you the actual results in graphic form, I just wanted to show you a couple of the cases of the elephants actually interacting with the experimental design. So I'm going to start playing. There isn't sound, but what you're going to see first is control. So this is just a string with some water on it. And what you're going to see second is the actual lion feces itself. So where we, again, scavenged fresh lion feces from the surrounding area, basically rubbed it onto a string and then stuck that string through a PVC pipe. So here you can see the control. The elephant is being asked to cross. He's kind of interested in the string. It's new. Sniffs it a bit. There's the trainer behind. There's one in front, and crosses no problem. And this is with the lion feces. You can see him smelling it. He's not so sure, but remember, he's being ordered. Oh, he's being ordered by 2 trainers. And I don't know if you can see in the bottom left here, you'll see oranges start to roll in more and more. And so here you can see what we interpret as a clear sign of hesitancy. The elephant backs up, more oranges are offered, more commands are given. And there's another orange, and the elephant is not having it, is not willing to cross over the string with the fresh lion feces, despite basically being offered an entire bag of oranges. You can see him there just giving up and moving on. Here, I want to show you what it looked like when we did the same thing, where we've got the control here with the pipe. So this is the PVC pipe with the water-soaked string going through it. And then followed by the PVC pipe, what you'll see is the indole and phenol, so the synthesized lion fecal volatiles. So here's the lion, new thing, sniff, doesn't care. Here's the indole and the phenol. Here he is sniffing it. And again, a bit of a hesitance response. Not totally sure. He's being urged by trainers behind him and in front of him. And finally does agree to cross, but after quite a hesitation response. The result, and it's important for me to note that N actually equals 4. So even though there were 5 elephants at this particular reserve, 1 elephant, after his first encounter with the lion feces on the string, refused to participate in any future trials. Absolutely would not go near that area again, and we weren't about to force them. So these results represent the 4 elephants that stayed and continued and interacted basically with all of the different conditions. And what you can see here basically is on the y-axis, the time that it took in seconds to cross. Again, if they refused to cross, we would basically code it at, I think, 180 seconds, or however many seconds are in 5 minutes exactly. And then here you can see the different controls. So we've got control with just the pipe, just water, control with water on a string, wildebeest feces, impala carcass, giraffe feces, dog feces, cheetah feces, lion feces. And then here on this side, you see the synthetic mixtures that we tried. So this was indole alone, again at 1 part per million, phenol alone at 1 part per million, and phenol and indole together. And what's important to note here, and what I want you to notice, are how things are grouped by color. So if these things are in yellow, they are not significantly different from the water control in terms of the time it took the elephant to cross. Those things that are marked in green were significantly different from both the lion and control. So that basically in this case is the dog and the indole and the phenol alone. And then the things marked in red, which in this case are cheetah, lion, and then phenol and indole together, are not significantly different from the elephant's response to lion dung. And so we've got a promising result here, well, here as well, that lion feces alone might be very effective, and that it might be possible to very cheaply synthesize the compounds in lion feces that are actually contributing to that deterrence effect. Now, if you look at it from the analysis from the point of view of the number of commands given, basically we have exactly the same thing, but instead on the y-axis, instead of time to cross, we have the number of commands given, and it's a bit more kooky and all over the place. But again, what you can see here is that the cheetah and the lion were not statistically significantly different from the phenol alone in this case, as well as the phenol in indole together. So again, compelling indication that lion feces is an effective deterrent, that cheetah feces might be as well, and that there's quite a bit of promise in terms of being able to potentially synthesize those relevant compounds. So real feces is certainly the best deterrent that we've discovered so far, but there's a couple of caveats, things that I want to note. So in our trials, and I've mentioned this a few times now, but it is important to note that we use ridiculously low concentrations of this stuff. Both the indole and the phenol were offered at 1 part per million. So it's possible that if we up the concentrations a little bit, we might actually see a similar response in terms of hesitancy from elephants as we see with the lion fecal samples, like the fresh lion feces. Another thing that we noticed, unfortunately, after our trials was that we were using equal amounts of indole and phenol, which isn't necessarily an appropriate signal, because consistently our chromatographs and those of other researchers who have done the GC mass spec of lion feces all consistently find that indole and phenol are actually present at quite a different ratio. So they tend to be present at an approximately 3-to-1 ratio in lion feces, whereas we were using a 1-to-1 ratio in our trials. So basically attempting to more closely mimic the lion feces by making a more appropriate ratio of these two compounds, as well as potentially upping the concentration of both compounds, might be as effective as the lion feces alone. And then the other thing is, at the very beginning, I talked about dimethyl disulfide. That was that third compound that was very commonly present in obligate carnivore feces. And because it smells really bad, and because we had to buy all of our compounds here in the United States and actually put them in an airplane to go to South Africa, because we couldn't set up receipts from Sigma Aldrich within South Africa, we just couldn't take dimethyl disulfide on the airplane, unfortunately. But it might be an interesting thing in the future to try. So in the future, what we're hoping to do is get back to South Africa, pandemic permitting, and try this again with higher concentrations of indole phenol, as well as the more accurate ratios, that 3-to-1 ratio I mentioned of indole and phenol. And potentially, if we can find a chemist to work with in South Africa who's able to receive things from Sigma Aldrich, potentially including dimethyl disulfide, which again is an important compound in lion feces. Another thing that we've started to work on is to really think about the elephant's landscape of fear. So for those of you unfamiliar with landscape of fear, this was new to me as well. It's basically a model based on the ecology of fear. And the idea is that animals behave differently. They use their spatial surroundings differently based on basically psychological maps of their habitats, right? Where they have in their minds areas that are more associated with the risk of predation, or signals that are most associated with the risk of predation in certain areas. And I think it'd be really, really interesting to look at elephants' landscape of fear. And we're in the final stages right now of completing an NSF to actually try to do this. And what we want to do is basically assess it using a model called the giving up density approach. And briefly, for those of you unfamiliar with it, what you do in this approach is you put a bunch of food out in areas where there is high and low predation risk, and that's predation risk that you were sort of inferring based on different landscape variables like distance to water or distance to villages, or how wooded an area is or how tall the grass is. And then you basically put out a favored food and you measure the density at which elephants give up feeding. So you can see in this little graphic here, this would be an example where this little bird has given up and left 3 seeds behind in this one blue area, and the bird has left 6 seeds behind in this red area. And the idea is basically that the bird was giving up sooner, was giving up at a higher density because of the risk of predation in this area. And so it's a really neat kind of way to directly measure and quantify basically the animal's perception of risk in its environment across different variables. So that's something that we're hoping to do. People have actually looked at this briefly before as part of giving up density and landscape of fear projects, looking primarily at ungulates in African savannas, and actually concluded that elephants don't have a landscape of fear because they weren't captured in their GUD experiments. What's interesting, though, is that their GUD experiments, being primarily aimed at ungulates, were very, very different, because what they were looking at in terms of landscape variables of interest were things like grass height, thicket density, etc. Basically things, landscape features that would obscure visual signals of their predators. Elephants have tiny little eyes, and I can't find any really good data on how well they can see, but they can't see very well. And so the idea basically is that maybe, previous studies didn't basically capture this potential landscape of fear and its effect on elephant movement across landscapes, because it was focused on environmental variables that had entirely to do with visual signals of predation as opposed to olfactory signals of predation. And so what we're hoping to do, here's some more clip art for you, is to develop a GUD model with olfactory predation cues that will look something like this: a bunch of their favorite fruit, marula maybe, or oranges, basically in some kind of up high basket, and allow the elephants in the wild, somewhere around Kruger National Park, we're thinking, to habituate to the experimental apparatus, and then sneak in some feces and sneak in some basically olfactory indicators of feces, and see how the elephants respond. And my ultimate goal is really to apply this. So I really, really would like, and there's so many reasons, it's a no-brainer to want to do everything you can to try to mitigate human-elephant conflict. I mean, obviously it has devastating financial and safety repercussions for people who live in countries alongside elephants, and it's also not great for the elephants either, right, due to retaliatory killings and basically the fact that they're driving the opposite of support for conservation schemes in these areas. And so what I'm hoping to eventually do with this, but I think I have to find someone who knows a thing or two about business and actual product manufacture, is to develop some kind of extremely safe, cheap product that can be used in the moment of conflict. And the model I have for this product is kind of shown here, because I'm originally from Canada, and it is sort of like a bear spray idea. For those of you who have not been lucky enough to spend lots of time in the backcountry where bears are endemic, this is a very commonly used thing that you would take out into the backcountry where bears are. And the idea is that if you encounter a bear, you basically spray this, and it will hopefully deter the bear and not just anger him. So that is it, actually. And I guess we can take questions. We've still got a bit of time.
Thank you very much, Kim. I think we can, as you say, open it up to the floor. If anybody has a question, I think they can ask directly. Maybe while they're getting their thoughts organized, I'll start off with one that I put in the chat halfway through when you spoke about the animal that flatly refused to continue in the trial. I'm quite interested in individual variability in terms of how animals respond to, be it climatic conditions or whatever stimulus. And so I was wondering if you had any further insights into that particular animal. Is it a different age and sex class? Do you have anything that you can pinpoint to try
That's
and
a really
explain?
that's a really good question, and not one, unfortunately, that I can answer. So it's Melissa that basically is the one that does the experimental trials with the elephants. So she knows more about the individuals. My understanding is that these individuals are all more or less the same age and that they're all adults. But it would be interesting to see if, maybe there was like a sex class difference or a slight age difference. But the point that you bring up is a very good one. People who study elephants are always very careful to point out that, there's a really big difference in the behavior and risk aversion levels of, say, a matriarchy-led group versus like an all-male band that might be, less likely to run away from stuff, be less neophobic. And I think that, again, because we only have these 5 semi-tame captive elephants to work with in the experiment, that's something we can hopefully get a better sense of doing this work out in the wild with actual wild elephants in Kruger.
Thank you. Is there anybody who would like to fire one off to Melissa?
Yeah, hi, this is, hi Martin, hi Kim, this is Claudia from Germany. I have one comment and one question. I find it really interesting how these elephants seem to be able to really distinguish between herbivore and carnivore scat, and it's a bit like sniffer dogs. So maybe you can also train sniffer elephants, just go around and I never would have thought that they are so good in distinguishing even between cheetah and lion. And the question is, do you think that they will eventually learn that it's only a smell and then afterwards will still cross the boundary they are not supposed to cross?
Yeah, that's a really, really great question. So habituation to any kind of deterrent stimulus is like definitely going to be a problem, I think, with anything that you try. The reason that we think that we're hopeful that these predator-based odors, that elephants might not habituate to them, is because as long as you are a wild elephant living in an area where there are obligate carnivores, where there are your predators, you should not ever be habituating to the smell of something that is associated with the most deadly thing in your environment. But we're not entirely sure.
How often do you think you have to refresh? Because I'm sure they know what old lion scat smells like compared to like really fresh one. If it's a week old or so, there's probably not a danger for them.
Yeah, I'm sure that they're able to assess the freshness of lion feces. I'd be surprised if they weren't able to. And so the idea isn't necessarily to, in terms of the application, the idea wouldn't necessarily be to like put these strings with fresh lion feces all around the perimeter of protected areas or around farms to protect them. The idea would more be to use these really cheap and in low concentrations, safe, commercially available volatiles, and to basically give people a can of them so that if the elephants show up, you can very quickly get out into your field and just start spraying odorants that they should be mortally afraid of.
Okay, thanks. I think it's very probable that you find a laboratory in Joburg or Pretoria which will be able to synthesize your dimethyl disulfoxide or whatever you were looking for, instead of you having to bring it on a plane.
Yes. It was also one of the, with fieldwork, it's always, you're super organized, and then in the last 24 hours, you find yourself ordering everything. So it was one of those as well. Yeah, we need to be better prepared.
Thanks, Claudia. Anybody else?
Hi, Kim. Kristen here. Thanks for a great talk. I have a quick question about whether you anticipate kind of trialing this with groups of animals compared to individuals, considering that, you know, vulnerability to things like predation could definitely relate to group size?
That's a really great question. So again, in terms of the experimental system that we have access to, it's not really something that we can do too much more with. Like, we really are limited to just 4 participating elephants. It's something that we're hoping to get a sense of, and certainly something that we're considering when designing like the details of the giving up density model, because you're absolutely right that larger group sizes should actually increase, it's going to increase the intra-specific competition at a feeding site, but it's also going to decrease the predation risk at a feeding site. And so that's going to certainly have non-random effects. So it's one of those things, unfortunately, because our experimental system won't necessarily allow us to get to it, that we're hoping to deal with it in Kruger, really, and hoping to get a big enough sample, enough participating elephants that we can actually say something about the effect of group composition and group size, and you know, how predation risk interacts with interspecific foraging to drive different strategies.
Yeah, that sounds very cool. Thanks. And I just had one quick comment that came to mind about the elephant spray, which sounds really intriguing. I just had one, you know, thought that came to mind is like here in the Western Serengeti, we have really large groups, cow-calf groups that raid crops. And, you know, with thinking about how close you might have to get to elephants, or a large group of possibly very upset elephants if they have been chased by people already, was just, you know, one consideration that came to mind.
Yeah, that's a really good point. I think that any product is going to, like, the creation of any kind of product or device or something is definitely something that we'd also have to trial in all sorts of different situations, because again, like, all male band of elephants is more likely to be aggressive, arguably, than a female, like a matriarchy led group with calves. There are so many potential confounds, and then you're absolutely right, the distance at which you have to be from the elephant to deploy a potential device like a bear spray type of thing is something we're going to have to think very carefully about as well.
Cool. Well, thanks so much for your talk. It was really interesting.
Thanks for coming.
Yeah, maybe a link to both Claudia and Kristen's question there. We often see at conferences and things that people come and speak about deterrents that they've tried, and you know, initially the results are quite good, but then with time, as Claudia alluded to, you know, the elephants get used to it, and I suppose part of that might be that it's takes quite a bit of maintenance to keep things going. And I was wondering if you had any opportunity, or somewhere in the back of your mind have played with the idea of maybe taking just the kind of thesis approach of some variation of that to a village in Uganda, say, and kind of seeing what happens.
Yeah, I would love to do that. I would love to do that. So we just actually finished this paper a couple months ago, and I'm stuck in America because of COVID. But yeah, I mean, there's so many cool things we could do, and I mean, my interest here, again, like my core research is very much about, you know, mutualisms, and doesn't really have to do with this. My interest here is primarily an applied one, an applied conservation thing. So I would love to get out there and start actually trialing things in the real world, and getting a sense of, you know, how do we get from, okay, we have some promising preliminary results about this, how do we get from that to something that's actually going to help people?
Yeah. No, we'll look out for that. Has anybody else? The floor is yours if you have a question.
No question.
Maybe maybe shows my ignorance. But what's the difference between the predator-specific odors and pheromones?
Pheromones are basically also VOCs, but they're meant for like intraspecific communication, but they can still be detected, I think, as volatiles.
Okay.
Yeah.
I don't see any other questions in the chat, and I don't see anybody with a burning question. So if there are no more, then Kim, I'd like to thank you for your time. Thank you for a very interesting presentation. Apologies again about the issue with time as we started, but that was really interesting, and thank you very much. And I'd encourage people to go and find the paper on the Conservation Science and Practice website. It's freely available, so you can simply download it there and have a look at that. And then I'd also just like to refer you once again to the SCB Emerging Issues in Conservation webpage, where you can see our upcoming schedule of talks, of papers featured in Conservation Science and Practice, Conservation Letters, and of course, Conservation Biology. So thank you very much, and good night or goodbye.
Thanks, Martin.